Okay, a slight variation in my title. Today I'll focus on what 12 years of monitoring has told us about baselines and drivers of coral disease on the Great Barrier Reef. And first up, let me acknowledge, like all big projects, this has been the collective effort of many outstanding collaborators and postgraduate students, whom I'll acknowledge as I go along and also list them at the end. Now, as we've been hearing over the past 1.5 days, and it has only been 1.5 days, seems much longer, the coral polyp is in the midst of a real David versus Goliath battle. In fact, over the millennia that corals have been building reefs, the puny coral polyp has been more than a match for all the natural disturbances that it's had to evolve with. But as we add to the mix climate change and various other human-related activities, we're perilously close to tipping corals over the edge to an alternative community type. So one of the biggest challenges for managers is to first understand the drivers of deteriorating coral health on the Great Barrier Reef, and then secondly, to manage them. And I'm sure most of you are familiar with this figure by Goliath et al., which shows that there's been something like 50% decline in coral cover over a recent 27-year period. In this study, they focused on crown of thorns, cyclones, and bleaching as the major drivers of this declining coral cover. But evidence has been emerging from other studies that diseases also contribute to the declining coral cover on the Great Barrier Reef. And in fact, in this study by Osborne et al., they found that coral diseases, in fact, contributed more to that declining coral cover, for example, than bleaching. And we think that, in fact, this still underestimates the role of disease in declining coral cover on the Great Barrier Reef. Now, I have to say that on the Great Barrier Reef, we're in a better position than our counterparts in the Caribbean, where there have been a number of outbreaks that have been decimating coral and sea fan populations over the last several decades, since the 1980s. So white band disease, black band disease, and a fungal disease. And too often we don't recognize the signs that disease tell us, the harbingers of deteriorating health, until diseases have reached outbreak proportions and populations have reached the point of no return. And the die-off of the diadema back in the 1980s in the Caribbean is a case in point. Even more recently, and alarmingly, since June 2013, there has been a major die-off of sea stars along the north coast of North America, along the whole coast of North America. And millions of sea stars and something like 20 species of sea stars have basically deteriorated into piles of slime and ossicles. It's probably potentially one of the worst marine wildlife epizootics that we have had to date. And in fact, just this morning, I received a report of someone had cited signs of this disease on sea stars just off the coast of Melbourne. So we're hoping we're not looking at a pandemic here. But in each of these cases, it's quite clear that we're looking at a disease epizootic. In other words, defined as a disease occurring at a time or place where it's not expected, or certainly at a rate greater than expected on past experience. Here on the Great Barrier Reef, we have identified 6 major diseases that do kill corals, but the conundrum is, what are the critical levels of those diseases that would indicate that that those levels are reaching an outbreak proportion? In order to be able to recognize those critical thresholds, which are needed for management, we need to have background knowledge on the types of levels of diseases that are typical for a given site. So that leads me to two of the major objectives for our research. First, can we identify baselines and outbreak thresholds for coral diseases on the Great Barrier Reef, and what are the drivers for these diseases? So we've been looking and monitoring coral diseases now since 2004, and we've been doing that on 3 sectors in the northern, central, and southern Great Barrier Reef, and at 3 cross-shelf locations. This shows you, for the northern sector, for example, reefs in the inner, the mid, and the outer continental shelf. We've been surveying the disease in 20 by 2 meter transects, and now have served well over 1 million corals. and 3 transects per site, 2 sites per reef, 3 reefs per shelf location, and 3 shelf locations per sector. Also, we have satellite-derived data for a range of environmental metrics at these same locations. So 3 metrics for sea surface temperatures, 4 for ocean color metrics that are proxies for water quality, wave height data that are proxies for storm and cyclone damage, and what we call other indicators of compromised health, notably predation caused by crown of thorns, starfish, and other predators, overgrowth by algal and cyanobacteria, bleaching, and so on. Now this shows you the patterns in that data over the 12-year period, first shown by sector in the southern, central, and northern sectors, and by shelf position in the inner, mid, and the outer shelf. And one of the first things that you notice is that overall, disease prevalence levels tend to be low, somewhere between 1% and 4% here on the Great Barrier Reef. But what I'd like to note is that even low levels of disease prevalence can equate to significant mortality through time. And just to show you the results of another study that we've done recently, where we tracked 50 colonies of corals that were tagged through time and measured rates of tissue loss. So here, this shows you the extent of the disease in March, then in April, and 2 months later in May. So overall, that's the rate of tissue loss in a 2-month period. And we calculated, based on those 50 tagged colonies, that even for levels of disease in the order of 1.5%, we would end up with a 2% loss in tissue over per month, which, when you add that up over a year, starts to become a significant amount of mortality. The other pattern that's been fairly consistent over the years is this greater disease prevalence on the outer shelf reefs compared to the inner shelf reefs. So we wanted to explore some of these patterns a little bit more. And what we did was we developed a Bayesian hierarchical model to compare disease prevalence across the shelf. And here I'd like to acknowledge Erin McNeil. So Erin is the modeler and the master data wrangler who's behind this analysis. What you're looking at here, this is a figure that summarizes quite a lot of data. It shows the posterior distributions for disease prevalence and compares them across the inner, mid, and the outer shelf. And we're looking at these distributions for 5 major diseases on the Great Barrier Reef. The open circles here represent the median expected prevalence. So these would be the expected values for disease at a given shelf location and region. The rectangles represent the 95th percentile of the posteriors, and these equate to unlikely levels of disease and background levels of disease. So in fact, this provides us with the capacity to define a threshold. So beyond those levels, those would be unlikely levels of disease based on our data. And then the solid black circles represent the 95th percentile of the data. So these are the extreme values. And you can see here they go up to 39 cases per 1,000 corals here for scleral eroding band. So with these 2 values, the 95th percentile, so this outbreak threshold, and the extremes in the data, we now have the capacity to scale the disease, the magnitude of an outbreak. So we have a threshold, and when disease prevalence are close to those thresholds, you would say sort of it's the low end of an outbreak, whereas if they approach these extremes, then we're getting up to extreme levels. And as well, here we're comparing the prevalence of disease across these 5 different major diseases, and we can see that scleral eroding band is by far the most prevalent disease on the Great Barrier Reef, followed by white syndromes, brown band, growth anomalies, and black band disease. And the patterns here, in general, for these first 3 diseases, have greater prevalence on the outer shelf reefs, but then for these bottom 2 diseases, they tend to have low prevalence, but those prevalence levels are consistent across the shelf. But importantly here, we now have a mechanism to try and identify baselines, thresholds, and of disease on the Great Barrier Reef. And these are shelf-specific for each disease. Then we looked at unraveling some of the drivers behind these levels of prevalence. And here we used 22 biophysical model-selected parameters. So these are the ones that had the greatest contribution to the levels of disease. And what we're looking at here are the standardized estimates for the contribution of each of these covariates to the prevalence of each of these diseases. Now, just to walk you through that a bit more in more detail, these black circles and lines, all right, these represent the median posterior values for the circles. The thin lines represent the 95% posterior densities, and the thick lines, the 50% posterior densities. So where these circles and lines are colored in dark, in black, they represent strong evidence of an effect of that particular covariate. In other words, the 95% confidence, 95% interval does not overlap 0. Where they're in this dark gray color, the circles and lines, that means the 50% interval does overlap 0. So that provides some evidence of an effect for that covariate. And then where they're white, that means that the 50% interval does overlap 0. So we have no evidence of an impact or an effect. If we go back to SEB, what this means is that we now have evidence that the percent cover of the acropora is an important driver for disease prevalence for prevalence of this particular disease. And that makes sense, given that we're finding it has highest levels of prevalence on the outer shelf reefs, and that's followed then by levels of branching parietes, and also other indicators of compromised health, notably cyanobacterial overgrowth and algal overgrowth. In cases here, these are wave heights either greater than 4 meters or 2 meters, which equate to whether we're looking at cyclone damage or severe storm damage. Here, we have lower levels of the disease when wave heights are greater than 4 meters, and that probably just means that there really are no corals left, or very few corals to have disease. And then this last block here, we're looking at temperature metrics, and what this is suggesting is that we have some evidence of an effect of a warm thermal anomaly, the mean positive summer anomaly here. But if you have a warm winter, then that reduces the disease. And that may potentially indicate that the coral host is reducing its susceptibility to the disease because of the more benign conditions over the winter. So basically, the beauty of this analysis is that for all of these diseases now, we have these standardized estimates, so we can compare the relative contribution of each of these drivers to the prevalence of the disease and really start to get to grips with what some of the drivers are of disease on the Great Barrier Reef. And lastly, I just wanted to end on with some results from a recent study which just provides further evidence of the importance of coming to grips with baselines and drivers of coral disease on the Great Barrier Reef. Here, we know that disease is a three-way interaction between the environment, the host, in this case the coral holobiont, and a pathogen. And climate change is affecting many things in the holobiont and in the pathogen, things like increased host susceptibility, decreased symbiodinium, but also changes like increased abundance and virulence of the pathogen. So we focused on these three factors and modeled what the risk of disease onset was with warming oceans if we took into account those three factors in particular. And here's a figure from a recent paper of a study led by Jeff Maynard, and it shows model projections for the latest year by which rising temperatures, and in this case we're looking at the representative concentration pathway is 8.5, which in fact is the one that's we're currently on. It's modeling what's happening at the present with emissions, and what it's showing, it's the year by which the thresholds for host susceptibility, pathogen abundance, and virulence will be exceeded here along the Great Barrier Reef. So all of these points here are colored in this region here, meaning that by the 2030s at the latest, we will be seeing disease outbreaks on the Great Barrier Reef. And if we do the same modeling for bleaching, what it's suggesting is that we won't be seeing the onset of annual severe bleaching events until sometime later in the 40s. And if you put the two together, then what it's suggesting is that there is a difference in the timing of the onset of disease outbreaks versus annual severe bleaching projections of somewhere between 20 and 30 years. So we're likely to see more mortality due to disease than bleaching in the next few decades. Now, that's not downplaying the importance of bleaching by any means. It's simply highlighting that disease is also a consideration that should be on our radar. And finally, just the closing comments. Disease prevalence, I'd like to point out, can be low in snapshot surveys, but nevertheless, mortality through time is high and significant. We now, from 12 years of data, can develop baselines, and from that we can identify thresholds that do signify an outbreaks and scale their magnitude, and we now can start to explore drivers of disease and how they vary amongst disease types. And as just shown, climate change-related factors are likely to increase coral susceptibility to disease, and so now when we model projections, there is significant disease risk in the coming decades. And just to thank an amazing team of excellent collaborators that have worked on these surveys for the last 12 years. Thank you.